as well as the transcribed mRNA. Now see here, polarity of this strand is not mentioned. Okay, we have to write down the polarity of the strand, as well as what we have to write. We have to write the transcribed RNA. Now see, we know that there are two strands. One is the sense strand. The sense strand is the non-template strand. So RNA do not synthesis over the template strand. Here we required for the synthesis of the RNA only and only with the antisense strand, that is our template strand, that is this one. Okay, so now here define the polarity of the two strand and transcribed mRNA. So only we have to transcribe the mRNA. So suppose if this is 5 prime, so this is going to be the 3 prime. So we will just write down antisense strand. Student, better I feel like that we should solve it into the Excel, so it will come one by one. Okay, so better we will solve it into the Excel only. Okay. Now what will be the sequence of the transcript? So here I will write 3 prime. Tell me the strand, that is G, then C, then T, A, C, sorry, P, G, C, T, then we have A, C, G, G, A, P. You also solve with me. T, T, T. Then we have G, V, C, P, G, then C, T, G. Just a minute, students. Now then we have C, T, G, C, T, E, C, G, G, E, T, T, G, C, T, G, C, T, G, G, C, T, and G, T, A, C, G, G, C, then B, A, G, E, A, C, C. Just cross check this time with the given question. Have you cross checked? Is it correct? Now this is going to be the 5 prime. Now we will write this is going to be the 5 prime. Now what will come with D? Now this will become very easy for us. Okay, now you tell me. Let me cross check. G, C, T, A, C, G G A T T G C T G P A C G C A G T A C C and 5 prime. Now I have written just antisense. You will also write the sense trend also. Okay, anti sense trend, or better you can write the template trend. What is asked? What will be the sequence of the transcribed RNA molecule from the given double-stranded DNA segment? Define the polarity. Now we had already defined the polarity. We have returned the antisense strands. Now only we have to write down the mRNA. Now tell me what would be the mRNA. Now see, this is mRNA. So we, in case of mRNA, we do not have T. Instead of T, we will be using the,
U.
yes, very good. C, G, A. Now what will come here?
U.
G. Then here C, C. Then here instead of T, U will come. Here A, A. Are you following how it is? We are changing how we are making. Should write it very neatly in the journal. Sorry. Here A should come. Now here U, because we do not have T in mRNA. Why this mRNA has gone exactly ulta? Okay. G. Now here what will come?
U.
Very good. Here
A.
now tell me here.
G.
Very good. Okay. Now that that is done, this is only asked. Write the mRNA. Shall we solve the next question? Now, third. Let me copy the third question first. Forget these words. It's very difficult. Now I will copy question here only. Okay, let it be. We can do here also. Determine the amino acid sequence of the peptide chain translated from the given transcribed mRNA. Now, what is given to us, children? We have got the mRNA. Student, here the image has cut from this side. Let me take the another screenshot of this. Okay. Now it's much better, I guess. Okay. Determine the amino acid sequence of the peptide chain. Now, what we have to determine? Amino acid sequence. Now, you should be ready with the amino acid chart to given you all. Now, see, we have learned that if we have got the mRNA, so what we have to write now, mRNA, we have the codons. So we have to change it with the anticodon. Now, this anticodon will not have any type of the polarity. Only we have the polarity with of this mRNA. So mRNA we have 5' to 3' polarity, and with mRNA's polarity we go and we write the tRNA codon. Then thereafter we will be writing the polypeptide chain. So shall we start? First we need to write down this amino acid sequence. Okay, 5'. Sorry. A student, if you will save, that will be become very easy for me to write. AUG. What is happening? A, then U, EG, G, G, then I have U, G, U, A. Student, why I have not written it earlier? Why I am doing with you all, so that you can also write it. Okay, do it with me. Then only you will be able to solve it in a better way. Okay. Then I have C, G, U, U, C, A, then G, G, then I have triple C, then U, then 1, 2, 3, 4, 5 times A, 5 times A, then C, then A, G, A, then I have 3 prime, right? Okay, this is the problem. Just a minute, student. Okay. Okay, let us solve this. Now, this is what? This is your mRNA. Now, students, we know that the mRNA has codon, right? Change क्यों हो जा रहा है again and again? This is mRNA codon. Okay. Now, what we will do first? First, we will be writing the anticodon. Now, see, students, when we have mRNA. So mRNA, it is nothing but the codons which we are getting from after the transcription. So after the translation, the tRNA adds the anticodon over it, and this with the help of that the polypeptide chains form. So this anticodon is already given, or you can say the codon chart I have already given to you all. So you all are supposed to open that chart. Now I will write the tRNA anticodon first. tRNA anticodon. Now for the just a minute, student. Yes, so tRNA synthesis 5 prime to 3 prime. So what will come here? Yes, come on fast. tRNA is again the RNA, so no T will be there. U, U, then here C will come, then here U, U, G, U, 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 you in theory in detail we all are going to talk about how all this happen, how these codons are added, all this interesting transcription and translation we all are going to learn in the theory. T, C, then we have U, U, G, A, A. Here what will come? C, G, A, 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 A. Then here U, A, C, A, C, C, sorry, and this is going to be the 3. But better not to write polarity in the case of the tRNA, or you can say the polypeptide. Usually the polypeptide, they are denoted with the C and. So the and with this we will be making 3 codon. So this is going to be one codon. This is going to be the one we will take, one will use. So I told you that 3 codon makes one amino acid. Now always synthesis you should remember is 5 prime to 3 prime. You might have observed that I have started writing from this side. 1, 2, 3 leaf, then 3 codons. This we will give the some other color. Okay, better to have this color. Synthesis is always in the 5 prime to 3 prime direction. You all are supposed to remember this thing. Now open your codon chart fast. Have you opened? Now tell me what is the code for UAC. Now how to read this codon chart I will tell you. Open your codon chart. Now see here first base is given. This is going to be your second base, and third base is always in the form of the UCAG. Okay, because this is UCAG. So first base is taken from here, like in genetics we do. So now tell me what is the code for UAC? What is the code for UAC?
Tyrosine.
What? Tyrosine. Very good. Now I will not write full tyrosine here. I will just write TYR. Okay. Now tell me what is the code for CAC. Come on fast. CAC is the histidine if I'm not wrong.
Histamine.
Yes. Next
Yes, ma'am.
AUA. AUA is your isoleucine. Right? Shall
Yes,
we go
ma'am.
ahead? Then triple A. What is your triple A? It is the lysine.
Glycine.
Then what is GCA? GCA is
Alanine.
alanine. AGU. What is your AGU?
Serine.
Student, AGU is your serine. UCC. UCC. Come on fast. Again, it is serine.
Serine.
So you should remember it is called as the degeneracy of the amino acid, that one amino acid may have more than one codon. Now see, there are 64 codon and we have only 20 amino acid, standard amino acid, which suits or fits in this particular codon. Okay. Now next is triple G, right?
Glycine.
GGG. What is that? Glycine. Very good. AUU. A.
Isoleucine.
Very good. Isoleucine. UUU.
Phenylamine.
What?
Phenylalanine.
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 Phenylalanine. Remember, phenylalanine, tryptophan, and tyrosine. These are the three aromatic amino acids. We all are going to discuss in detail in the theory. Then next is GUU.
Valine.
Valine.
Valine. Very good. CUU. CUU.
Leucine.
Leucine. Very good. So this is the answer. This is our polypeptide chain. Okay. I hope you all have solved it with me. Chalo, shall we solve the next? Let us solve the next problem. This is very interesting to solve. Now question number 4. Enlist a possible amino acid would be replace leucine if one of the base in its codon mutates at one time. Now first of all, till I copy this, you find the code for leucine fast. Tell me the code for the leucine. What is the code for the leucine? That is UAA, right? UA, sorry, UUA
UUA.
and UUG. See, student, you check the page number 66 where I have given you the code, all the possible codes for the amino acid. So how many codes are the UUA? Then we have the UUG. What other codes we have for the leucine?
CUA.
CUA.
CUA. Very good. Then what else we have?
CUC.
C.
CUC. Very good. Then
CUG.
CUG. Very good. CUG. Then
CUU.
very good.
W.
CUU. Now see. How many codes are there? 1, 2, 3, 4, 5, 6. Now see, enlist the possible amino acid that would replace leucine if one of its base in the codon is mutated. Okay, now here let me clear that this is a leucine, codes for leucine. Okay, leucine codes. Now here one of its base is mutate. So if u u a, instead of a, what we will get? We will say mutation means the changes. So instead of a, if c is there, so what will form? What is the uuc, children? Come on, fast, find out what is the uuc. What will,
Phenylalanine.
what? Phenylalanine. So it will convert into the phenylalanine. Now you if it will have, instead of c, it will be having the mutation UUG. So what will form? UUG. Come on, tell me. What is UUG?
Leucine only, ma'am.
Very good. So that means it is not the mutation. A we have, okay, if it is U, then what it will be?
Phenylalanine.
Have you checked it
Phenylalanine.
properly? Yes, it is phenylalanine. Okay, so this is the mutation. This is again, this is not a mutation. This will remain the leucine itself. So this is a mutation. Yes. Now here instead of UUG, U U A will there. Then what will happen? It is going to remain the leucine only, right? Right or wrong? Again, if it is UUG, we have solved for this. We have solved for remaining all UUA and all and all. So no need to solve again. Now here CUA. So CUA. So instead of A, if U will come, so what is the CUU? Again, it is the leucine only. So there is no such mutation. Okay. Now here CU. G, what will be the amino acid? It is going to be leucine only. Right or wrong?
Yes, ma'am.
A हो गया. Now C. What is the CUC? Again, it is going to be leucine. So there will not be any type of the mutation. Come to the CUC. So here if CUA will come, what will it be? It is going to be the leucine only. We have solved remaining all, right? Here we have solved for rest all. So no need to solve again. Then CU. Come on, tell me. We have solved all this CUU, CUG, so no need to solve all that. Okay. So this is the solution for this question. So here blue marking is showing the this. If so, only one base mutation, and that too on the first base mutation is there in the case of the leucine. So there is a very rare chance that it is going to change the polypeptide. Understood? So you will write this into the conclusion. If there is only single base mutation into the first base of the leucine molecule, there is a very rare chance of the mutation because leucine has 6 codons. Okay, have you copied this thing? That is called as the Wobble. Give the possible anticodons on the tRNA molecule that would be engaged in the translation of mRNA into hexa. Okay, sure. Hubble hypothesis consider करना है. What is the Hubble hypothesis? Hubble hypothesis say A bind with the T, T bind with the C. C bind with the G. This is called as the Hubble hypothesis. Now see, give all possible anticodon on the tRNA molecule that would be engaged in translation of MLGFPM. Now this MLGFPM, this is a polypeptide chain is given. So open the page number 66 where I have given the amino acid and their symbol as well as the codon. Have you opened? Now polypeptide, be very careful while writing this polypeptide. Polypeptide M. M is for what? Your methionine. Methionine is always the first codon. Okay. So this gives us idea that this is the 5 prime. Okay, because methionine is always the first codon. So what is the code for methionine? There is only and only one code for the methionine, that is AUG. Okay, and this is known as the initiator sequence. Come on, what is the next? L. What is the L symbol for? It is for the leucine. What are the codes for the leucine, children? Now there are 6 codes for the leucine. I will copy from here only. I will copy these codes for the leucine. All 6 code for the leucine can come. Okay. Now what is the next? G. What is the G for? G is for the glycine. Okay. So what are the codes for glycine? These are the G, G, A. Also we have what we have glycine के लिए G, G, C, G, C, then G, G, G. G, C, G, G, A, G, G, C,
GGU.
and also we have one G, G, U, G, G, and U, right? Okay. Then next is your P. What is P for? Proline. So what are the codes for proline? We have C. Let me check the proline. CCA. Yes. Next, what we have? Which you tell? So that will be better for me. Then we have triple C. Yes. Next, what we have? Fast.
CCG,
Yes. C. C. G. Then
CCU,
CCA.
CCU. CCA we have already written, na beta? C. C. U. Okay, got it? Now proline is done. What we will do, we will give one color to one amino acid. So that will be better. Okay. Then what is the next amino acid, children? It is proline के बाद में क्या है? F. What F stands for? It is for the phenylalanine. Come on, tell me phenylalanine. That is going to be U, U, C, then triple U, right?
UUU,
Then what is the next amino acid? It is the again M methionine, only one code. That is AUG. let us fill this also the yellow color, so that would be better. There are many ways to make this particular amino acid, but we have written six possible ways, as we have the leucine with the six codes. Okay, there are many other ways. Now see, let us complete the other amino acid also. Will not give the polarity because it is not asked. So AUG will be common because methionine have the same code, right? So it will be common here also, here also. AUG is going to be common. Now here also these codes, any can come, but you can write this also. Here also anyone can come. Just a minute. We will write this. Now here anyone can come. Here it is give all possible ways. So all possible ways will be many. Because of this, there are many other ways. So these are the some possible ways. You can find many other ways also for writing this particular amino acid. You can change all this amino acid. You can make various combination because six codons are there. So these six codons are 6 into 4. So there are many possible ways. How to find out this possible ways? See, methionine के 6 codons are there. Okay. How many codons are there for for your this leucine? You have the 6 codon. Then you have the 4 codons for next two. So this way you can imagine so much big number will come and so much possible ways are there. So we have written few of the possible ways that are sufficient. Okay. Let us solve the next question. We'll be taking just one more question. Thereafter we'll stop. You might be having the next lecture now. This is a big question. We'll do it into the next class. Okay. Do you have next lecture? Yes.
Botany.
Okay, then we can stop now.
मैम जर्नल में भी ऐसे कलर करना है
You can for better understanding.